Welcome back, ladies and gentlemen, to the second of these talks on cancer molecular biology, in which I'd like to review the field of cancer chemotherapy, that is, the development and the application of drugs for the treatment of cancers. And to do that, I'll take a number of examples that over the last couple of decades or so have illustrated both the great advances and some of the setbacks in this field. And then I'll look at one or two other innovative approaches to this problem that I think might play an important role over the next few years. But before I do that, I'd like to talk for a few moments about what I sometimes call the greatest revolution. And that is the project to sequence completely the human genome that was begun in the late 1980s and completed in 2003. And this project, I think, arguably has had the greatest impact on medical science of anything in history. And so you might ask, well, what has it done for cancer? And in the limit, what it's done is shown that the enormous complexity of cancer by revealing the fact that if you look within an individual tumour, each cell at the level of the sequence in its genome is different to every other cell in that tumour. And that complexity has highlighted why we need drugs to be able to treat the almost limitless combination of genetic changes, mutations that can drive cancers. We'll look then at what we've got in the drug cabinet, and then we'll also look at some of the innovative approaches that I just mentioned to this problem of cancer chemotherapy. Not the least remarkable thing about the Human Genome Project was that it was completed in under the scheduled time and below the original budget cost. And two of the reasons for that triumph can be seen in this graph. On the left-hand side, the fact that the cost of sequencing per base dropped precipitously over the time course of the project, and the speed with which DNA sequencing could be accomplished rose correspondingly. And even more remarkably, that trend has continued since the completion of the project, such that now the cost of sequencing DNA has dropped by well over 4 orders of magnitude from what it was originally at the start of the project, whilst the speed has risen by over 10 to the 8-fold. Now we can carry out over 100 million sequencing reactions simultaneously in a technology called massively parallel sequencing. And the upshot of all of this is that it's led to the almost inconceivable project of being able to sequence all eukaryotes on the planet over a period of 10 years. And that project has just been launched under the title of the Earth Biogenome Project. But if we return to cancers, we've already mentioned that this sequencing power has been able to reveal the complexity of cancers down to the difference between individual cells. And in so doing, what that has allowed the sequencing to achieve is to compile complete mutational signatures for all the major cancers. And of course, in doing this, new cancer genes are being revealed all the time. And as each of these comes to light, they are themselves, by definition, a novel target for drug therapy. So the notion is that we may have entered a new era. That is to say that when a patient presents with a cancer, that cancer will be sequenced. That will give the tumour mutation profile. From that, we can dissect the key cancer drivers. We can even use mouse models, mouse avatars that include these cancer genes and test the effects of drugs on them before we treat a human being with the appropriate drug. And of course, the upshot of that is that it will lead to tumour remission. And if the worst comes to the worst, and in due course the tumour recurs, then we simply repeat the sequence. Now, the first evidence that we might indeed be entering a new era came hard on the heels of the completion of the Human Genome Project. And it was carried out at the Sanger Centre, just down the road from Cambridge, that had been the site of the British contribution to the Human Genome Project. And it was done by Mike Stratton and his group. And they examined a number of cancers using exactly the same technology as had been used in sequencing the genome. And from these, they came up with a really amazing result concerning the condition of malignant melanoma. Now, malignant melanoma, as you probably know, arises from cells in the skin. These are melanocytes, which produce a brown pigment, that's melanin, and that's the substance that gives skin its colour. Malignant melanoma is the least common form of skin cancer, but it is the most deadly. And of course, one of the causes of malignant melanoma is exposure to UV radiation. So perhaps surprisingly, it's a significant cancer in the United Kingdom, nearly 18,000 cases a year at the moment, and nearly 3,000 deaths. And in particular, it afflicts young people between the ages of about 15 and 35. And for them, it's actually the second most common cancer. You might guess that in Australia, the situation is even worse given the amount of sunshine they get. And indeed, there typically per annum, there are about 14,000 new cases, 1,600 deaths. It's the fourth most common cancer, and it's the most common one amongst young people, sometimes known, therefore, as Australia's national cancer. And it arises, of course, when some abnormality in the skin, the sort of thing that we normally think of as a mole or a birthmark, and that for the most part will never cause us any harm at all, starts to behave abnormally. That's to say, it starts to grow, change colour, starts to become itchy or bleed. And as soon as it does that, you should seek the attentions of a physician, because you have an incipient melanoma, and that at that stage in the proceedings, before it penetrates the underlying basal membrane, it can be treated by surgery with essentially 100% success. The mutation that was identified by Mike Stratton's group in this condition concerned a signalling pathway within cells about which we'd known absolutely nothing at all in cancer terms up until that time. Just a recap to remind you that cells are designed to be essentially impermeable to everything, and the way that they have of getting both nutrients and information across the outer plasma membrane is by means of specific membrane receptors that span the membrane. And these provide a site on the outside of the cell for signaling molecules such as hormones to interact. And when that happens, the receptor changes its shape, changes its conformation, and when the inner bit of the receptor changes its conformation, it can then set off a signaling relay of proteins that convey the information that the extracellular signal has arrived to the nucleus, and where they turn on transcription of specific genes that can drive proliferation. There are, of course, as I'm sure you know, other signaling molecules that can diffuse across the membrane. Those are the steroid hormones, but we're not concerned with them at the moment. The mutation that was identified by Mike Stratton's group was within one of these signaling relay pathways. It concerned a protein called BRAF, that is a serine-threonine kinase, and the single point mutation transformed that into a constitutively active kinase. So now the signaling activity was permanently turned on, the proliferation response in the nucleus was permanently turned on, and all of this means that the cell has now been rendered insensate to extracellular signals. It's a perfect example of the first of the Hanahan and Weinberg conditions that I mentioned in the first lecture. Remarkably, within just a few years of the discovery of the mutation in BRAF in malignant melanoma, a small molecule inhibitor specific for that mutation in BRAF had been developed, and it became in 2011 the first example of a drug designed by fragment-based lead discovery, also known as fragment-based drug discovery, to gain regulatory approval by the FDA, the US Food and Drug Administration for the treatment of late-stage melanoma, and its effects were indeed very dramatic. This is a 38-year-old man with this condition, and just a few weeks' treatment with vemurafenib had removed all evidence of the cancer. Despite this success, vemurafenib has turned out to be a very good illustration of the problems that continue to beset cancer chemotherapy, because within a short space of time, resistance to the action of the drug had developed and the condition had returned in essentially the original form. It's worth emphasizing that in this case we're looking at something that is a consequence of the extraordinary flexibility and adaptability of the cell. The drug has continued to work, and it's still inhibiting mutant forms of BRAF. But what we now know is that the cell has been able to activate a whole range of mechanisms to short-circuit this inhibition, from using other cellular signaling pathways to developing secondary mutations in BRAF that prevent the drug acting. So perhaps rather than a new era, we're actually seeing the problems that have confronted cancer for many years. We can indeed now sequence, but we may not be able to identify all the critical drivers. We actually still have few specific drugs for treatment, and in most cases, and we'll see other examples of this, tumour remission is brief and recurrence is inevitably. And that arises from the complexity of the genetic mayhem that we've mentioned in tumours. And I'd just like to show one experiment to illustrate just how complex this problem is. And there are many other examples now to illustrate exactly this from a variety of primary tumours. And this just is a cartoon illustrating the spread of secondaries to form metastatic sites from a primary tumour in the kidney. And in this experiment, not merely were the metastases examined by sequencing, but small biopsies were taken from different positions in the tumour. That's R1, R2, and so on. And when the sequence data was compiled, the upshot is something that bears a striking resemblance to an evolutionary family tree. This is actually drawn to scale in terms of the number of mutations acquired, and it makes the point that cancers originate from a mutation in a normal cell, and they then accumulate hundreds or thousands or tens of thousands of mutations. And you can see in this family tree the extraordinary differences between the different regions taken from the primary tumour, the different biopsies, and indeed from one of the metastases. So all of that brings us to the issue of metastasis, and that is, of course, a critical matter in cancer because it's responsible for the vast majority of cancer deaths. And as we've already seen in the cartoons in the first lecture, it arises when tumour cells can escape the primary site and intravasate into the circulation, and then reverse that process, lodge in a new site and colonise it. We know that the synthesis pattern of proteins in tumour cells changes for them to make their escape from the primary. And it changes again when they colonise a secondary site. But despite much effort in searching for genetic changes, none have been discovered that associate with metastasis. That's to say, there's no set of mutations that you can ascribe to the phenomenon of metastasis. It's inefficient. Most tumour cells never form a colony. Only a very small fraction manage to do so. And their development, again as we've seen, is supported by the recruitment of normal cells and of the vasculature of the host to supply the growing tumour with oxygen. This brings us to the tumour microenvironment, which is probably the hottest topic in current cancer biology research. And it's really the phenomenon that we're now beginning to understand in some detail, by which, as tumours start to grow, they act as an attractant for other cells from the host. So, in other words, they send out chemical signals, and as a result of these chemical signals, a whole variety of cells that include fibroblasts, for example, the connective tissue cells, and also the cells of the immune system, the lymphocyte family, macrophages and others, are drawn to the site of the tumour. Their initial efforts are directed to eliminating the tumour, because it's seen as an abnormal growth. But if the tumour can escape those initial efforts and begin to flourish, something extraordinary then happens, because these cells that have arrived with the intention of eliminating the tumour are now switched in terms of their activity. They release chemical signals that now promote tumour growth and indeed promote angiogenesis and further metastasis. Until relatively recently, we've known next to nothing about the molecular and cellular events involved in metastasis, but this is beginning to change, thanks in no small part to the efforts of David Leiden and his colleagues and of John Kondelis and his group. What I'm going to show now is a 3D animation made by his group showing metastatic invasion inside living breast cancer tumours in mice, based on their live imaging of fluorescent cells. The mouse tumour is actually laid on the stage of a fluorescence microscope to do this. The tumour mass is green, the cyan-coloured cells are macrophages, and the purple rods are collagen fibres. And we'll see a cell break away from the tumour mass, migrate to a collagen fibre, and use this as a sort of tramway to get to a blood vessel. One of the cyan macrophages follows suit and migrates to the fibre and moves along it in step with the tumour cell and actually signals to it as they proceed, indicated by the flashes. When the pair reach the outer surface of the vessel, it is enzymes from the macrophage that produce a pore in the blood vessel, enabling the tumour cell to pass through and enter the circulation of the host. This work is clearly producing new targets for chemotherapy, drug targets for treating metastasis, and indeed drugs are the only way that we can treat metastases, with the exception of using radionuclides for bone metastases. And the drugs we have available are small molecule inhibitors, monoclonal antibodies, and then the field of immunotherapy and virotherapy, and I'll say a little bit about each of these in turn. There are hundreds of cancer drugs. Over 400 combinations have FDA approval, but very few of these are actually specific. This list of specific inhibitors is not comprehensive, although there aren't that many. We've already met one example, vemurafenib, used against mutant BRAF in melanoma, and you may have come across erlotinib and gefitinib that target specific mutations in the epidermal growth factor receptor and in the Janus kinase 2 that occur in non-small cell lung carcinoma. But even when these treatments are initially successful, patients frequently relapse, and this has led to the development of second-generation drugs directed against mutant forms of the EGF receptor. Imatinib was the first protein kinase inhibitor to receive FDA approval, and it targets the BCR-ABL fusion kinase that we met in the first talk, and is used to treat chronic myelogenous leukemia and acute lymphocytic leukemias that are Philadelphia chromosome positive. And AG221 is a recent addition, an inhibitor of mutant forms of the isocitrate dehydrogenase 2 enzyme that occur in acute myeloid leukemia. Two examples of monoclonal antibodies are Herceptin, which is relatively well known as a treatment for breast cancer when the HER2 receptor is overexpressed, and Avastin, an anti-angiogenic agent that targets VEGF-A, one of the vascular endothelial growth factors. This was approved in 2004 for the treatment of breast cancer, but in 2011 it was withdrawn because cumulative evidence indicated that it had very little beneficial effect, and indeed, when patients relapsed after treatment, the returning cancer often appeared to be more aggressive than the original tumour. And the third category includes checkpoint blockade, gene therapy and virotherapy, and we'll turn to those now. To summarise very briefly the normal immune response, a cytotoxic T cell, that's a type of lymphocyte, also known as a killer T cell or a cytolytic T cell or a CD8 positive T cell, kills cancer cells, interacting via its T cell receptor with an antigenic peptide attached to a major histocompatibility complex molecule on the surface of the target cell, and it then releases enzymes that trigger apoptosis in the target cells. However, there are a number of immune checkpoints, regulators of the immune system, and they're crucial for self-tolerance. So they prevent the immune system from attacking cells indiscriminately and giving rise to autoimmunity, diseases such as psoriasis or rheumatoid arthritis. But some cancers can protect themselves from attack by stimulating immune checkpoint targets, and this has prompted the development of monoclonal antibodies to block checkpoints. And the currently approved checkpoint inhibitors block CTLA-4 or PD-1 and PD-L1. So PD-1 is a programmed cell death protein 1, and it has two ligands, PD-L1 and PD-L2. And the rest of this slide simply makes the point that the basis of checkpoint blockade is to use specific monoclonal antibodies that interact with these proteins that are responsible for downregulating the normal immune response. That's to say, anti-PD-1 antibodies or anti-PD-L1 antibodies or anti-CTLA-4. And there are now a considerable number of ongoing trials, notably combining the use of checkpoint blockade with small molecule inhibitors such as vemurafenib. And I've just showed some data from one of these that is really rather typical of the current picture. So this is actually using an anti-CTLA-4 monoclonal, which is a way of boosting the immune response, as we've seen. But the problem with these sort of trials so far has been that a number of patients don't respond. Some respond so badly that they have to be withdrawn from the trial. And in those that do respond, the overall survival increase is not that great. We're not really looking for increases of the order of a few months when it comes to cancer therapy. All of which suggests that we don't really know enough about the tumour microenvironment that we're trying to regulate. The next method of immunotherapy that I'd like to say a little bit about is adoptive cell transfer immunotherapy. And the basic method picks up a weakness in the patient's immune system, whereby it makes lymphocytes that kill tumour cells, but it can't make enough. Their protective effect is overwhelmed by the growing cancer, and by taking small pieces of surgically removed tumours and growing them in the lab, it's possible to select those T cells that have killing capacity, and then these are expanded over a few weeks to make enough cells to keep on growing when they're infused back into the patient. And the upshot is a hefty boost for the natural anti-tumour defence system. And the pioneer of this method, called adoptive cell therapy, is Stephen Rosenberg, who worked for many years at the National Cancer Institute in Bethesda. A wonderful and perhaps unforeseen benefit of this method is its efficacy against metastases. And it's perhaps not surprising, as it's what Rosenberg calls a living treatment. In other words, it just gives a helping hand to what nature is already trying to do. A more sophisticated approach to boosting innate immunity is to introduce new genes into the genetic material of T cells to target them to tumour cells with greater efficiency. Then an ordinary blood sample suffices as a starting point from which T cells are isolated. And one way of getting them to take up novel genes uses viruses, essentially just genetic material wrapped in an envelope, and the virus is disabled so that it has none of its original disease-causing capacity, but retains infectivity. It sticks to cells. The method of redirecting T cells to a desired target was pioneered by Gideon Gross and colleagues at the Weizmann Institute in the late 1980s, and it has since led to sensational results in treating relapsed B-cell acute lymphoblastic leukemia. To the fore have been Renier Brenchens and his group from the Memorial Sloan Kettering Cancer Center in New York, and the genetic modification they used made the patient's T cells express an artificial receptor on their surface called a chimeric antigen receptor, or CAR. This T cell receptor was designed to stick specifically to a protein known to be displayed on the surface of the leukemic cells. That's CD19. And in this slide, you can see on the top left-hand side a typical stained view of a blood sample from a leukemic patient with a large number of very dense purpley stained cells. But after a very short treatment with the modified T cells, those cells have essentially completely disappeared and the patient is to all intents and purposes in remission. Now, the first success with this method was in a child with ALL in 2012, and since then CAR T-cell therapy has seemed to be a safe treatment, and some 70 to 80% of patients appear to achieve complete remission. However, as more long-term follow-up data are published, it seems that there's a high risk of relapse after CD19 CAR T-cell therapy, perhaps as high as 50%, and so major problems remain, even though this has been a startling advance so far. To illustrate virotherapy, I've used TVEC as an example. The virus is herpes simplex, the relatively harmless cause of cold sores and genital herpes. And in this construct, two viral genes were removed, one that shuts down an individual cell's defences, and another that helps the virus evade the immune system. And a gene for a white blood cell growth factor was added, human granulocyte macrophage colony stimulating factor. The viral drug works by replicating in cancer cells, causing them to burst, and the inclusion of GM-CSF stimulates an immune response against the cancer. The final 4-year analysis of the phase 3 study that led to FDA approval for TVEC showed a 30% response rate with a 17% cure rate, and overall there were significant survival benefits in patients with early metastatic disease and in patients who hadn't received prior treatment for melanoma. TVEC was the first oncolytic immunotherapy approved globally. That was in 2015. And although, as these data show, it's not perfect, it does illustrate the potential of this approach. And the reference is a review of the field that gives a more complete picture. Having considered a number of chemotherapy approaches in some detail, I'd like to step back and look for a moment at the broad picture as an introduction first to an alternative strategy, and then in the last part of the talk to new methods for screening and monitoring that are referred to as liquid biopsies. Now, we've seen that a whole range of chemicals in the external environment, mitogens, cytokines and so forth, can signal to cells, and aside from the steroid hormones, they communicate via transmembrane receptors that in turn talk to the nucleus via multiple relays of proteins. And in principle, mutations in any of the components can constitute a cancer driver. And I actually find it easier to think of this as a collection of clouds. And the idea behind personal medicine is to produce drugs specific for a given mutation or mutations in an individual cancer somewhere within one or more of these clouds. Thus far, this has not been as successful as anticipated, prompting the notion that as all these signals are converging on the nucleus to activate genes associated with a cancer phenotype, there may be a central gateway or portal through which all these signals pass, and such a node might be a good target for a pan-cancer inhibitor. Impersonal medicine, if you like. A major candidate is MYC, a master transcription factor that is essential to drive cells through the cell cycle. In mouse models, blockade of MYC can not only stop tumour growth, but can reverse it, so that the tumours actually disappear. You can see this on this slide, which shows a lung tumour stained with H&E, and all the dark purple masses are tumour clones of cells. But if MYC is blocked, that not merely stops tumour growth, but the tumours actually disappear. Now, in these experiments, MYC is regulated genetically, so the details don't matter here, but you can consult the reference if you're interested. If different combinations of driver cancer genes are used, the mouse tumours turn out to resemble the human varieties that they model, but their pathology looks the same independent of the mutations that cause them. But they differ between tissues, lung versus breast, for example, and that's been the basis of clinical classification until sequence signatures began to take over. But that means that the tumour microenvironment is critical, and it's recently emerged that MYC controls this too. These are immunohistologically stained sections of lung tumours driven by oncogenic RAS. And on the left, in what is essentially normal tissue, the lung cell marker shows up as green, and red is a cytokine called interleukin 23, and that's in the tumour microenvironment. What does IL-23 do? Well, it's a pro-inflammatory and angiogenesis promoting cytokine. So what is extraordinary is that after 3 days of switching MYC on to give abnormal expression of MYC, this tumour microenvironment has been invaded by cells releasing interleukin 23. Thus far, blocking MYC activity in these mouse experiments has, perhaps surprisingly, not had any significant adverse effects. And so the notion of a pan-cancer treatment targeting MYC is alive and well. Watch this space. The last therapeutic approach I'd like to mention takes advantage of the fact that cells shed DNA into the circulation. Tumour cells do this too, and individual cells also break away from the primary and pass into the circulation. Modern technologies using oligonucleotides and antibodies permit these very minor components to be isolated from just a few mils of blood. They can then be sequenced, and from that it's possible to define the cancer site of origin. And the hope is that this will lead to much earlier detection. But what are called liquid biopsies are now being used to monitor response to chemotherapy, and I'll show two examples. The first, a vemurafenib type treatment followed by immunotherapy, and you can see that the amount of circulating mutant BRAF reflects the response, an early effect that was not sustained in this case. However, in another patient, successive courses of immunotherapy lead to sustained tumour suppression. A very recent addition to this story has come from David Leiden's group. A few years ago, they had shown that tumour cells and cells in the tumour microenvironment release small vesicles called exosomes that carry proteins and also nucleic acids. And these tag landing sites for tumour cells when they are released into the circulation. The astonishing discovery that a pre-metastatic niche is formed even before tumour cells leave the primary. They've now shown that in a variation on the liquid biopsy theme, it's possible to isolate these exosomes from a small blood sample, and analysis of their contents permits cancer detection with high specificity, to the extent that they think this method will become a screening standard within a few years. Well, ladies and gentlemen, I think that might be a good place to pause and to summarise what we've described. The immense impact of DNA sequencing on cancer over the last 15 years has revealed a genomic landscape that is heterogeneous even down to the level of individual cells within a tumour. This is a frightening prospect from a therapy viewpoint, but what it has done and is continuing to do is offer multiple targets, and in seizing upon these it has given rise to a huge expansion in chemotherapy options, and that includes the new fields of immunotherapy and virotherapy. And with this has come the concept of personal medicine. And whilst we've seen some examples of success, there are clearly many problems to be solved before this concept becomes widely applicable and has the desired outcomes. It's therefore worth considering other approaches, and the notion of a pan-cancer method remains attractive. Recently, variations on the liquid biopsy theme have already given advances in response monitoring, and there is strong evidence to suggest that these may transform detection, advancing the stage at which cancers can be first detected, not just by months or years, but perhaps by decades. And that's exciting because we know that the sooner a cancer can be treated, the more likely is a successful outcome. And finally, may I thank you all for listening to these talks, and I hope they have been helpful. It is for me a great disappointment that I cannot this year meet the members of the M.Phil class in person. Nevertheless, I can still wish you all possible success and happiness in your scientific career ahead. And once again, thank you very much for listening.